Well, and welcome to the Merv Griffin Radio Hour. No, it's just Chris and yet another one of his wacky theme songs, which can only mean one thing. It's time for yet another Ball Publishing webinar. I'm Chris Beytes, editor of GrowerTalks magazine and Green Profit magazine and the e-newsletter Acres Online, and I'll be your host for the next 50 minutes or so as we tackle today's topic. Well, it's right there on your screen: mites in the nursery, identification, miticides, and biological controls. Now, we are going to get to the bottom of these annoying invertebrate arthropods, and to do that, we need an expert because, well, as always, we know it's not me. I'm like you. I battle spider mites, but I am not an expert in them. Who is? Well, he's from Clemson University in South Carolina. He's Associate Professor of Entomology, Soils, and Plant Sciences. He is Dr. J.C. Chong. JC, are you there, my friend?
I am, Chris.
Beautiful. Now, where are you broadcasting from today?
Oh, Chris, I'm broadcasting from my office in Florence, South Carolina.
Very nice. But you were someplace else just this last week. Where was that?
I spent 2 weeks in Sicily.
Sicily.
Looking for awesome.
Looking for mites, no doubt.
No, actually, I was attending a conference on scale insects and other crazy whackers we have to deal with.
Well, that's excellent. And what sort of a mite season have we had so far in 2016?
Well, it has been somewhat of a quiet one, but it kind of depends on what is going to happen this summer. If we're going to have a dry summer, oh, the mites are going to come rolling back.
Oh, well, you can only hope, right? Because I know there's nobody more disappointed than an entomologist who can't find a bug on a plant.
Well, we're just going to be out of a job. Come on now.
That'll never happen to you.
No.
And, of course, I am your host, and I'm broadcasting, as usual, from the windowless office right next to a storage closet in the Ball Publishing broadcast studios just west of Chicago. Now, a very special thanks to our generous sponsor, BASF, who made this and our other BASF free webinars possible. In fact, they're going to be doing one more with us later on in July. Now, I'm going to pop this one up for those who aren't hearing me right now. If you can hear me, you can ignore this. But if you can't hear me, well, I hope you're reading it and you're figuring out which to click on to get some audio. But I think everybody's getting me now, and if not, they'll follow that little path there. One other little note here: if you have questions as we go along for Dr. Chong, use the little chat area that you see over there on your control panel. Just type it in, and if it's pertinent to what we're talking to at the time, I'll ask the question, and if it's not, we'll save it for the end. And if we run out of time, well, I'm going to give you Dr. Chong's personal email address and his private cell phone line. No, I won't do that. So he can answer those questions for you, and I know he'd be happy to. And lastly, if something goes wrong, or something goes right, maybe a customer comes in, or you can't stick around for the end of the webinar, fear not. It will be archived at the usual spot, ballpublishing.com/webinars. It's the same place where you sign up for them and find all of our other archives. It'll be up within, well, it won't be up quite as quickly as usual because Jen Zerko, who's my manager of all things internet, is the one who archives these, and she's out today, but we'll get it up as quick as we can. So that said, JC, are you set to go to tell us all about mites?
I am.
All right, take it away.
Let's do this. Good morning, good afternoon, everybody. Thanks for joining this webinar. We will, as Chris already mentioned, talk about mites today. I have given this same talk to different audience, but never to a national audience online, so this is the first.
Right. This could be international.
Well, who knows? Who knows? But either way, I'm very excited. So, for those of you that had attended my seminar in Oregon, California, Texas, or Louisiana before, well, think of this as a review and an opportunity to stump me with questions you have carefully considered over the past few months. If you have any questions, again, please type in your questions in the message box, and Chris will direct my attention to them. So let's get started. But anyway, so whenever we look at a nursery, that's a perfect picture we want to look at. Plants are healthy, everything is nice. Now, you know this doesn't last long forever. Sometimes some kind of insects will come in and kind of try to eat into your profit. Of course, we got the mites. For those of us working in the nursery in the southeast, ambrosia beetle is always a big problem. For those in the northeast, and Japanese beetles who might come along, caterpillars might come along, white flies, scales, aphids, thrips, you name it, all kinds of stuff. So all these pests, we need some kind of tools to deal with them, to control them, and whatever tools you use would have to deal with what kind of insects or what kind of pests you're dealing with. If you got a big grasshoppers, a 22 is your solution. But next, please, for a little mite like this, you just might have to use something else. So don't fear not. We got a lot of solutions against mites in ornamental plants. And today, what we're going to talk about really is about identification, life cycle, monitoring, controls, cultural, biological, chemical, and let me say something. I have prepared a awesome handout for everybody, but I'm going to make you work for it. So if you want a handout that covers everything I talk about today, please send me an email, and I'll pop that handout to you immediately. I will have my email address at the end of the presentation, so you kind of just have to stick around. Sorry, I'm not going to give it to you right now. All right, next. So there are quite a few mites out there, and in fact, mites, there are more species of mites than birds and fish and plants combined. There's that many of them. And the biggest family that we typically deal with in ornamental production are the spider mites. This includes quite a few different species. And for those of you that work in the greenhouse quite a bit, you probably remember broad mites. For those of you who work with African violet, cyclone mite. And those two are actually part of the family of mite, what we call the thread-footed mite or tarsonemid mites. Eriophyid mites is becoming more and more of a problem, particularly for plants that's grown outdoor. And for folks in Florida, they would have to deal with some of the other pest flat mites or false spider mite, like the red palm mite that is causing quite a bit of issues on the coconut palm in Miami at the moment. Next, please. So, how do they feed? The mites do not feed like typical insects. When you think about typical insects like caterpillars, they feed by having these two chomping mandibles. They just kind of chew and tear into plant tissues. But for mites, it's very different. So they sort of have this wedge in front of their head. They use this wedge to just sort of insert themselves into the plant tissue and then sucks up all the plant juice. From there, that would include all the cell content. Next, please. So where on the leaf, when the cell is completely empty, all you have left is basically the cell wall on the outside. The cell wall actually looks like silver or white in some way. That's why a very typical damage symptoms of spider mite is actually that silver stipplings. So each one of those stipplings is an empty plant cell. So when you have a lot of them, you're going to have a lot of problems. Now, one of the biggest problems we have when it comes to ornamental production in either greenhouse or nursery is the two-spotted spider mite. And two-spotted spider mite is by far the most common species that we deal with. They are actually quite easy to control. As you probably know, they are tiny, much smaller than a pinhead. And if you look at a picture on your far right corner, you're going to see a male, a female, and the eggs of a two-spotted spider mite. Typically, the female is more rounded on the rear end, and the males are more pointed on the rear ends. And the eggs are actually quite big. So typically, spider mites do not produce a lot of eggs per day, but over a long period of time, it's quite possible that they can produce several dozens of eggs over their short life period, about 7 days or so. As you can see, the two-spotted spider mites cause that silver stipplings on the foliage most of the time. Next, please. And the reason why they're called spider mites is because they spin silk. And in this case, you can see that the silk just completely covers some of your plants. And when you have a plant that's completely covered with silk like this, don't even think about controlling them. By this time, it's already too late. Just the best way to deal with that is probably a trash bag. Throw everything in there, tidy it up, and throw it away. Now,
that's enough to give any grower the willies right there.
oh, it definitely will. And to make you even more grows out, if you look at this picture more carefully, you're going to see that there's some kind of brownish bits on the web on that marigold. And those brownie bits usually just kind of move around quite a bit. Those are actually balls of spider mite waiting to hitchhike on you or got blown off by the wind to the next plant. Maybe that other marigold in the background. So very quickly, you're going to have a spider mite spreading by either wind or on our clothing or on our tools through the nursery or the greenhouse, and you've got a infestation going on very quickly. Next, please. Another species of spider mite that look very similar to a two-spotted spider mite is the Lewis mite. Now, the Lewis mite, identifying Lewis mite from spider mite is not that easy based on the appearance. Now, the two-spotted spider mite typically have 2 spots on the back, and the Lewis mite has 4. But there's quite a bit of variation there. Lewis mite typically is a problem on poinsettia grown in the greenhouse. On the outdoor, I don't see them very often at all. So poinsettia, if you grow poinsettia, Lewis mite could be a huge issue. Next, please. Boxwood spider mite is another common species that we have in the southeast. And this particular spider mite feed on boxwood alone and also cause their stipplings as well. And if you look at them, they're usually sort of brownish and darkish brownish gray color. Next, please. And all the other mite species that I just talked about, two-spotted spider mite, Lewis mite, boxwood spider mite, all of those spider mites are what we call warm season mite, meaning that their population will grow up, will become built up in late spring and really become a big problem in the summer when it's warm. That's why they're called the warm season mite. Now, the next 2 species that I'm going to talk about, they're also spider mite, but they are called as cool season mite, meaning that the population actually builds up in the spring and fall, and they just kind of gone diapause or quiescence during the summer. So it's a little bit different. So if you see any kind of damage on your plant in the summer, it's possible it's two-spotted spider mite or one of the warm season mite. But if you start seeing damage on your plants in late spring or early summer, then it's quite possible it's one of the cool season mite. There are 2 very common cool season mite species we have. One of them is the southern red mite shown on the screen. They're bright brick red color, feeding on the underside of the leaves, very easy to identify. 2 azalea, camellia, and hollies are the 3 most common species I find them on, at least in South Carolina. And again, they will cause their stipplings on your leaves. Continuing feeding is going to cause that bronzing color on the leaves as well. Next. The next species of a cool season mite is the spruce spider mite. Spruce spider mite is a specialist of conifers. Seen them a lot on arborvitaes and junipers and such. And they're usually kind of dark greenish color and start feeding along the ribs of the needles. And you're going to see stipplings showing up on the needles. Next, please. And let's talk about something else other than spider mites. The thread-footed mite or broad mite was an old enemy, especially for those of us that work in the greenhouse quite a bit. Now, what they do is that they will feed on the growing tips of the flowers and cause that kind of distortion as shown on the pictures here. Years and years ago, this is a very big problem for greenhouse growers, but no longer. We are starting to see more and more of a broad mite infestation in the field nursery as well. Why? Well, we don't really know. It might have something to do with the environmental changes, or maybe raising humidity and such. We don't know for sure. But it's very easy to identify broad mite. If you have a good hand lens, take a look at the populations, especially those eggs. Those eggs are very unique. If you look at them, they got little pimples on the top. Make them look almost like a football under your hand lens. Next, please. The next species of the tarsoneme mite is cyclone mite. Cyclone mite used to be a huge problem for African violet growers, but now they have become somewhat of a minor pest, and we only have occasional infestation here and there. Again, they cause distortion on the growing tips, and as seen on this picture of African violet. Next, please. The next group of mine is becoming more and more of a problem, especially those folks that work in the nursery and landscape as well. One of the biggest issues we have in the southeast right now is the rose rosette mite. Some of you probably heard of that before. Rose rosette virus transmitted by the rose rosette mite to a lot of different rose species and cause a very typical distortion and multiplication of the terminals as seen on this picture. In fact, the infestation is so bad around here that a lot of plantings actually stop growing roses, or that they put up all the infested roses that they have. At this point, we do not have a very, very good management approach to control the rose rosette virus and the rose rosette mites. My colleagues at Tennessee are working on that. I hope they can come up with some solutions soon. Next, please. Another species of Eriophyid mite is the privet rust mite. And this, as shown in that picture there, you can kind of see them crawling around a little bit. One thing I forgot to mention about the Eriophyid mite is that they are not difficult to identify. They are very, very small. Typical hand lens might not be sufficient to see them. But once you see them, you know it's an Eriophyid mite because their body is elongated, looks almost like a banana, and only have 2 pairs of legs. So they would just kind of crawl around. They cause that kind of distortion and cuppings on the flowers and the leaves. And for the privet rust mite, they will feed on the young leaves and cause a downward cupping of the foliage. An occasional problem, but it is a problem nonetheless. Next, please. A lot of Eriophyid mites actually cause gall on different kind of landscape trees and shrubs. And this is one of the gall species, the maple bladder gall. You can find them on just about any species of maple anywhere in the U.S. You got that little reddish little pimples on the leaf surface. If you open it up, you're going to see inside almost look like a tumor, and that's what Eriophyid might typically do to a plant species. When they are feeding, they either inject a toxin or virus into the plant tissue and cause this multiplication of cell division. And that almost causes a gall, or basically you can think of that as a tumor on the plant. And the mites basically feed within that gall, protected from the environment. So it's actually a very interesting surviving strategy. Now, of course, interesting from an entomologist's point of view. For those of you guys out there that have to management, they could be a really pain in the rear end. Next, please. So now that you kind of know about the mite species, let's talk about how you can actually find them. So looking for the symptoms is probably the best way to actually figure out whether you have a mite problem or not. Now, that kind of stipplings and that sort of discoloration on the foliage are actually very good sign to tell you that you have a problem. For us, who are the growers, we look at the plants day in and day out. We know what they look like. So if there's any kind of symptoms that look like that, take a closer look. Look at the bottom side of the leaves and see if you can find any kind of mites at all.
JC, before we get too far, Luke wants to know, what about the aloe mite?
Aloe mite. Luke, I have no information about that. Unfortunately, it is not something that I typically deal with over here, but I can do some research and get back with you.
You don't think, do you know that it's its own species of mite and not just another one that you've already gone through that happens to attack aloe? It is a specific
I have heard of aloe mite before. In fact, I heard it out in California. And aloe mite is a Eriophyid mite, and what they will do is that they'll attack the growing tips. Same thing, very similar to all the other Eriophyid mite, which they attack the growing tips and the flower buds of the aloe and cause that tumor-looking growth on that. It's very difficult to control. I'm not very familiar with this particular species, but I can, like I said, I can do some research on that and find out what needs to be known.
All right, thank you very much.
All right, so again, let's look at the symptoms again. For the Eriophyid mites, similar to the aloe mites, there's going to be some sort of distortion. On the top, That is, I believe, a maple, something similar. And what you can see that is that after feeding, the plant cell basically just divide uncontrollably, and you have this growth or this tumor growing on it. And the aloe might create something very similar to that. Next, please. So another way to monitor for your mite population is to do, a beat sheet method. It's very simple to do. You can get a clipboard, put a piece of white paper on top of it, put that clipboard underneath the branch you want to sample, just whack the branch. Hit everything in it, make everything drop onto the white paper, then you can use a hand lens to take a look at the white paper and see what fall off. Now, you're going to have all kinds of stuff. You're going to have all kinds of debris, all kinds of insects, and all kinds of mites as well. So if you use this particular method, how do you know it is a mite that you need to be concerned with, or is it a beneficial mite? It's actually pretty easy to tell. If it is an herbivorous mite, meaning that the phytophagus where they're feeding on the plant, typically they're kind of greenish, yellowish, pinkish, and they walk really, really slow. Their food source is right there. There's no hurry. There's no reason for them to be hurried to go anywhere to look for food. Now, if it is a beneficial mite or predatory mite, they are typically pinkish, orangey, or somewhat reddish, and they usually have got long legs and walk really, really fast. They walk fast so they can catch up to the other mite species and eat them. So by observing for their behavior, you can actually tell whether it's a phytophagus mite or whether it's a predatory mite. Pretty easy to do it. Next, please. So hand lens. I mentioned hand lens a few times. That is most important tools you can have when it comes to pest management. Mites are small. You need something to help you see, and you can use that for some other pest species as well. So a little typical hand lens at the 10 times magnification like that costs, say you bet, maybe $10. If you buy a plastic one, maybe $2. So it's a cheap tool, but it's going to help you a lot down the road. Next, please. So let's talk about control. There are quite a few ways we can control spider mites. The first thing I want to mention is quarantine. So most of the plant material we have in the nursery and as well as in the greenhouse comes from somewhere else. Now, if you have a chance to actually quarantine the plant's material that's coming in, you can actually have a chance to stop any kind of invasion by all these pest species. Just set them away in the corner of the nursery. Just look at them for a few days. If they're starting to see some kind of symptoms showing up, control them if you want to keep the plant material, or send them back to your supplier and ask for a refund. Now, but in the greenhouses, I have a joke coming with this picture, I think. Let me think about that. Oh, yeah. If in the greenhouses, a lot of times you ended up having grandma's plants inside the greenhouse with you. Try not to do that. You never know what kind of pest species might be on grandma's plant. As much as you love your grandma, do not take her plants and put it in your greenhouse. If you do that, you are somewhat introducing problems into your greenhouse as well. Next, please. Sanitation is also very important. If you have a plant that's completely covered with spider mite, throw it out, because sometimes it will cost you more to control them than to get rid of plant material that are infested. If the plant material are only few and far between, this is a very good way to get rid of your infestation. If you have a much more extensive infestation, you probably need to think about applying a miticide or insecticide to control the population. Next, please. Water and humidity can also be used to control spider mite populations, two-spider spider mite especially. Now, most of the spider mites and two-spider spider mite as well, they prefer to have a dry and hot environment. When it's dry, when it's hot, the development really speeds up, and also the reproduction will speed up. So you actually have a bigger infestation. So in a greenhouse, if there's any way to control the humidity at all, if you keep the humidity high enough, the spider mite population typically is lower. Next, In the nursery, that would be a little bit more difficult to do. There's quite a bit of anecdotal information that shows that spider mite population or infestation seems to be more severe along an access road where it's dusty and dry. So along those roads, one way you could probably reduce the spider mite infestation is to actually spray the plants a little bit more often, especially the foliage. You spray it, increase the humidity, you will reduce the population growth. But of course, you need to kind of balance it with the potential of developing more foliar diseases. So it's a very fine balance, and you just need to kind of play with it a little bit and then figure out what would be the best balance for your particular situation. Next, please. All right, so biological control is becoming more and more practice by a lot of growers, particularly in the greenhouses. In the handout that I prepared, I have several tables. One of them is on predatory mites and insects that can be used to control different mite species. Over here are some of the examples. Let's see. All right, so Ambrosius cucumulus is really used to control thrips, but they could also feed on cyclone mite and broad mites. Neosillus californicus is probably one of the more commonly used ones, and is used mostly to control spider mite, but it can also feed on cyclone mite and broad mite, and it could feed on thrips as well. The most commonly used predatory mite species is actually Phytoceus basimilus. This is probably the number one predatory mite for spider mite. Quite a few of those are being used in the greenhouse to control them. But in the nursery, typical nursery situation, Neosillus felicis is actually the one that is being used more often than Basimilus or Californicus. You could certainly use Californicus or Basimilus, but they seem to work much better in the greenhouse, in my opinion. The next few, one of them is a predatory midges of the spider mite. Stetorus, that's a lady beetles, tiny lady beetles that would feed on spider mite. And you have entomopathogenic fungi, for example, Bovida bassiana, that you can just mix in water and then spray just like an insecticide to control spider mite, broad mite, Eriophyid mites, thrips, whiteflies, and all kinds of insects. So there are quite a few biological control options that's available commercially. Next, please. So here are some of the examples that I've shown you earlier. Phytoceus basimilus, that is the most common predatory mite for controlling spider mite. This particular species really do well if you have a warm environment. In a cooler environment or cooler months, a lot of folks actually use Neosillus californicus on the top. And the predatory midge, Acarisugi, is also a very good biological control to deal with spider mite as well. Next, please. Now, I always say that biological control is really a good control method against spider mite, but it is probably best used as a preventive control or against a low pest infestation, meaning that you probably should apply biological control agents as almost like an insurance program, whether you have a low population or no population of spider mite. So if you basically what you would do is to do periodical releases, just release it very on a calendar basis, just to keep the population down. Biological control cannot be used for curative control. It's very difficult and it's very, very expensive. If you have a spider mite population that's so big that it's already causing crop damage and that biological control cannot really do the job, you might need to use some kind of miticide. But if you are going to use miticide, there are quite a few questions it seems you need to be considering. One of them is, how close are you to shipping? If you are going to be close enough to shipping, miticide really is a good solution for you because it can really knock the population down. Biological control works great, but you would need to give it a little bit more time. That's why it's more of a preventive control rather than curative control. And also, the second thing is, what kind of miticide do you have? Do you have something that that is compatible with biological control agents? Or if you're not, what are you going to do? So that's another question you need to consider. And also, we just go back to the compatibility issue is, if you are going to use a miticide or insecticide, what would it do to your biological control agent? And that would really determine what kind of miticide you're going to be using. We'll talk about that in a little bit. Next, please. But
JC, before we go on, we have a couple of questions that are related to biocontrols. Robert wants to know, earlier you had, in using the white paper test, you mentioned beneficial mites have long legs and they move quickly. He wants to know, are there any conditions in which a pest mite would be moving quickly across the plant surface, such as warmer weather or longer days?
That's a very good question, Robert. Yes, if the temperature is high enough, the pest mite will also move a little bit faster, but it's never going to move as fast as a predatory mite. So it's hard for me to give you a sense of how fast it might be, but if you put them side by side together, you're going to be able to tell the difference. Again, the best way to do the identification would be to take a look at them with your hand lens.
Yeah, it probably takes some experience. You know, you need to look at enough mites to get to know what does a regular mite move at, and you know what's fast, what's slow. Jim wants to know, same topic. If you have a large amount of predatory mites or beneficial mites out in your crop, is that a sign that there's a large population of the pest mites that they're feeding on?
Well, Jim, let me give you a thought. Well, that could mean, to me, that could mean two things. The first thing is that you had a large population of pest mite previously, so that the predatory mite actually feed on those pest mite and build up their population. It's possible that if you see a lot of predatory mite, it might be at the end of the pest mite population, because the predatory mite already built up to an extent that you can find quite a few of them. Yeah. Another way I can look at it is that yes, it could be that you have a lot of pest mite, but if you're going to find a predatory mite, you're going to find a pest mite right next to them. So if you find a lot of predatory mite, and if the reason is that you have a big pest mite population, you will also find a lot of pest mite.
And probably a lot of symptoms on your crop as well.
Absolutely.
Yeah. All right. Excellent.
All right. So, there's a lot of things we need to think about when you're practicing biological control. I always say that biological control agents are living organisms. They have their own requirement as far as what kind of condition they prefer, what kind of pest species they would prefer to feed on. So biological control is a very cerebral exercise. You need to be thinking a lot about your production practices and see how you can actually use a biological control. When it's done well, biological control works perfectly, beautifully, and you can do a lot of works with biological control. But one biological control program developed for one particular situation is not necessarily transferable to another production system. Look at the kind of production system that we have in greenhouse, nursery, and landscape. In the nursery, for example, you have containerized plants, shrubby stuff. You could containerize woody plants, or you have field-grown woody plants. All these three different production systems would actually give you a different environmental conditions, and because of these different environmental conditions and practices, you actually will have to adopt your biological control program to fit these specific conditions so that it works the best. So next, please. So there's really not a single recipe that I can develop for you, and then you can use it for every single one. So every time you develop a biocontrol agent, biological control program, it is best that you think about the entire production system holistically and design one that fit your particular production system. Next, please.
JC, you don't have shade house on there, and Vicki wants to know, is a shade house indoor or outdoor?
Oh, shade house, is it indoor?
Talk about a cerebral problem there.
Well, actually, that is more of a EPA problem, I guess. No, this is the kind of questions that we extension agents, extension entomologists always have to think about. Is this, well, is it indoor or outdoor? But most of the time, for me personally, I consider a shade house a outdoor structures. They are not completely enclosed within four walls. So I would consider a shade house more or less a outdoor nursery production.
Outdoor, but we're on the safe side if you do that. I think, yeah.
Yeah, probably. And the best way to figure it out is probably talk to your pesticide regulator people. They might have a different definition from me. In fact, I probably got one or two email in my mailbox right now from these folks and say, you were wrong. They were indoor. Who knows?
Another quick question. Do growers typically stick with one predatory mite, or do they switch to cold or hot environment mites?
Yes, they do not. Well, actually, kind of come out a little bit strong here. Most of the growers will change the program depending on the season. And a lot of greenhouses, if they're growing crops in the warmer season, Persimmons seems to be the one that's preferred. But when it comes to a cooler month, when they're still growing some kind of cooler season crops, California has become much more suitable predatory mite species to use. So it does change. Very similar to the message that I'm trying to convey in this particular slide, which is you have to tailor your biological control program depending on your production system.
All right, here's a question for someone who I know is very into biocontrols. He wants to know about Lamonica mite and when it will be available in the U.S.
Lamonica mite. Well, I heard that before, but I cannot tell you when that's going to become available.
But that's a new beneficial mite used in Europe, I would assume.
Let me look at that. Yes, that would be one of the covert biological system have I believe it's covert. Let me go back now.
We'll check that one out and
we'll come back and let me check it out and we'll come back to that.
All right. And then lastly, before we go on, what about rainy and dry season for predatory mite selection? Any preferences?
Rainy or dry season for predatory mite selections? Well, I have not seen a huge difference between the two. So I would not say, in my opinion, in my experience, there's not a whole lot of differences between the two.
All right, we'll keep moving here.
All right, so as I said earlier, that all the biological system would have to be very different depending on what you're dealing with. And the best people to talk to about it, fortunately, is not me. As you can tell, I do not really know a whole lot about what is coming down the pipeline, and I know what I know, then that's all there is. And the best people to talk to, in fact, are the folks that will provide you with all the biological control agents. And this would be the guys that produce and sell them. They would include Biobass or Syngenta Bioline, Cobert or IPM Lab, BSF, working with Nematode, and BioWorks got quite a few products as far as entomopathogenic fungi is concerned. So there's quite a few suppliers that they have. Now talk to these folks because they know best what they can offer you, and they know best when to use their product, and they can also talk to you in more details about your particular production system and figure out the best way to actually develop a program for your productions. And because this is their business, they want to make sure that you are very successful when using their product. So they want to make sure that you get everything that you need to make it a successful program. So, and also if you need to look at suppliers for all the other natural enemies, you can check out this particular publication from California. All right, so there are quite a few ways you can apply biological control agents. In the shade house, in the greenhouses, for predatory mite production dispersal, you can use some kind of brand a lot of predatory might actually come in little jars full of brand, and you can just sprinkle them on top of the foliage. And another way you could do that is do a sachet, for example, the one on the corner there. What it would do is that the little predatory mite will come out from the sachet slowly, a few at a time, and they will slowly spread out. Another way, if you are mechanically inclined, you can always make a sort of big old blowers and blow all the brand into the ranges. But of course, there are some other smaller equipments that you can use to help spread the mites in the greenhouse or the shade house as well. Next, please. And in the outdoor situation, biological control could be done, and in fact, there are a lot of study being done right now to find a way to actually figure out how can you enhance the environment so that you can attract and conserve the natural enemies in your particular system. And that has been done a lot in the outdoor production. The idea is if you can plant some kind of flowers in the middle of the rows, one thing is to stop erosion and moderate the temperature. Another way is if you pick the right plant species, it could attract natural enemies, which would feed on the nectars and the pollen that's provided by the plant. When they are done, they can spread onto your crops and control the pest species for you. Now, at this point, I would say that this particular method, it sounds great, but there's still a lot of kinks that we need to be working out. For example, what particular plant species you need to be using so that you can attract the right species of, say, predators to your particular nursery. So that's something that we're still working on and trying to figure it out. Next, please. But the one thing that you can do now to really help with biological control in the outdoor situation is to use your insecticide and your miticide more judiciously. Insecticide is designed to kill. That's what they do. So inevitably, they are going to have quite a bit of impact on the biological control agents that either you release or attract to the nursery. Next, please.
This question on biocontrol, before we move away from it, JC, is Tim wants to know if any of the predators that you've shown control russet mites.
The russet mite, well, the information that I have on the, let me find that, my handout. The handout that I mentioned earlier actually have that particular information on that. The russet mite actually is one of the Eriophyid mites, and that could be controlled with a few of these predatory mite species. One of them is Ambrosius andersoni, ANDRSONI, Ambrosius andersoni, and that particular species could be used to control russet mite or rust mite. And another species, let me take a look. And that's about it. That would be the only species on my list that I know of that have been used to control russet mite or spite.
All right. Well, Tim, stick on and get that that address that you need to be able to get that handout, so you'll have it.
All right. So let's talk about miticide. There's quite a few miticide that's available out there. Some of them just very generally say mite and do not provide you other information as to whether it's spider mite that they control, or broad mite, or two spider or Eriophyid mite or russet mite that they control. So some of the miticide is kind of iffy. Information is kind of iffy. Some of them are very, very specific. Again, there's quite a few of them, and on the list here, I have another good list on my handout. So make sure you get it from me as well. And there's quite a few of them, and here are some of the lists of the miticide I have. Starting with carbaryl. Carbaryl 7. We use that. It's an oldies and a goodie, and we use that quite a bit. And it has control mites in general. Not a whole lot of information. And all the way down to phenosicarp or precrude, and contos or spirotetramat. Spirotetramat also have control for mite species, as well as horticultural oil and insecticidal soap. Now, there's quite a few of them in here. Next, please. But I would recommend that you do not use this particular chemical. If you would go back one slide, Chris. So let me explain why I think you shouldn't be using them. A lot of them, even though they have mites on the label, I have not find them to be very, very effective in controlling mites. For example, bifenthrin, it has mites on the label, but I have tried several times to use bifenthrin to control mite species, spider mites especially. It doesn't work as well as I would hope it would. And there are some other chemical that I would use it for something else. For example, spinosad. Spinosad or conserve has been, for me, one of the best product to control thrips. So, but some of the resistant issues are starting to pop up with spinosad. So I want to make sure that I don't overuse spinosad. And so I'm going to keep spinosad to control thrips instead of using it for mites. And another thing would be Cantos. Cantos or spirotetramat, it has activity against mite, but I'm not going to use it to control mites. Cantos is a systemic insecticide. What if one day that we are going to lose neonicotinoid, and Cantos is going to become one of the few alternative we have for neonicotinoid. So I'm just going to use Cantos or skip it for sap feeding insects control. So these are not some of the insecticides that I will use to control mites. Next, please. One more. All right, there we go. And no fear, though, because there are a lot of miticide out there that you could use to control spider mites or any kind of Eriophyid mites or broad mite as well. You have 1, 2, 3, 4, 5, 6, 7, 8, 9 different mode of action of miticide out there to control spider mite. So it's not very difficult to find one that would fit your particular use site requirement, and also one that you could mix and match with the other mode of action to develop a miticide rotation program. You have something that is could be used for nursery, greenhouse, and landscape. Something that could be used for only nursery, only greenhouse. So there's all different kind of combination. There's a lot of miticide that is that you could use, that you can select from for your particular situation. Next, please. But which one do you want to choose really depends on what you're going to use them for. Miticides are not created equal. What miticide you use really depends first on what you're trying to control. Is it against spider mite, against tarsal mites, or is it against Eriophyid mites? So identification of mite is very important. Some of the miticides have very broad spectrum activity. For example, avid, it will control aphids. It will control spider mites and control tarsal mites. It will control Eriophyid mites. Some of them have very broad, very narrow spectrum of activity. For example, Sultan, which is a new product by BASF, it will only control spider mites. So if you have a Eriophyid mite infestation, obviously Sultan would not be one of the choice that you'll be using. Next, please. And what miticide you choose also depends on what particular mode of action that it is. And of course, that's signaled by the IRAC group number on the label. And this is very important when you try to develop a miticide rotation program. And so every time you make an application against a mite species, you want to follow up with another product of a different mode of action. Next, please. And it also depends on what particular mode of exposure that miticide is. There are two different modes of exposure for of miticide. One of them is translaminar. The other one is contact. Contact is relatively easy to understand. Contact is basically miticide that will kill mite by direct contact with the mites, or when the mites actually walk across the residual and get in contact and got killed that way. Translaminar miticide, on the other hand, is somewhat, you can think of it somewhat as a locally systemic. If you spray the miticide on the upper surface of the leaf, the active ingredient actually will penetrate the leaf tissue and kill the mite that is feeding on the underside of the leaves. But translaminar doesn't mean systemic. The translaminar miticide will go from the upper surface to the lower surface, but it doesn't go into the phloem and got translocated to the entire plant. Now, there are different species, avid, for example, has both translaminar activity and contact activity. A sultan has only contact activity. Next, please. And what miticide you use also depends on what life stages you are dealing with, whether it's eggs, whether it's nymphs, or whether it's adult. Avid has activity against immatures and adult. Sand mite has activity against eggs and immatures. And sultan has activity against all life stages. So if you have a population that's mostly adult, you could use sultan and get rid of all the life stages, or you can go with avid. So that would kind of depends on what life stages you ended up having. Next, please. And also, different residual longevity. Most of the miticide have 28 days residual longevity, but that longevity really depends on the environmental condition. But typically, I would recommend that your reapplication do not be more than 2 weeks. So basically, reapply every 2 weeks. That's because most of the miticide are really protectants. Whether they are contact or translaminar miticide, they will protect leaf tissue that is present during the time of the spray. Any new tissue that develop after the spray are not protected. So you want to make sure these new tissues are also protected as well. So I would say, even though some of the miticide, for example, sand mite has a 45 days residual, I'm not going to wait 45 days before I apply it. I will probably just apply it every 2 weeks or so. Next, please. And also, miticides are not created equal because of the different compatibility with biological control agents. Some of the miticides are very compatible. For example, sultan in some of my tests is quite compatible with most of the predatory mite species that we deal with. And also, but if you're dealing with avid, which is somewhat compatible, then you might have to think about what is the best way you can use biocontrol agents and spray avid at the same time, and not end up killing your biological control agents. So there's going to be a lot of thinking about a lot of development of trying to think about the best way to reduce your impact on the biocontrol agents if you're going to use a non-compatible miticide. Next, please. So there are a lot of biocontrol, a lot of miticide out there, and you're going to have a lot of options when it comes to developing a miticide rotation program. Next. So there are some guidelines as far as rotating your miticide. Miticide rotation is very, very important because spider mites have only a 1 to 2 weeks life cycle, and they can develop miticide resistance very, very quickly. So you want to make sure that every spray you have should be a different mode of action. And if you are going to tank mix different miticide in the same tank, you want to make sure they are different mode of action as well, or different IRAC number. Next, please. So let's talk about, here are some of the examples that I come up with that just to give you some example how you can rotate miticide. For example, if you have a light infestation of spider mite and most of them are adult, and that sort of signal the early stage of the infestation, these are probably on your new cuttings where you have a few adult mites coming in through your seedlings and whatnot, and you're at the very beginning of the stage of the infestation. So I would probably go in with avid, or if you're in the greenhouse, you can have pylon and give a quick knockdown of the adult, just wipe off the adult, and then follow up with a miticide that affect all life stages, so that you can clean up the adults and clean up the eggs that might be produced by those adults. After that, you can just follow up with just about any kind of miticide you have, just to keep the population down. Next, please. Here's another example. This is dealing with a moderate level of infestation, and in this stage, you have different life stages. You got adults, you got eggs, you got some immatures as well. So this is basically the middle stage of the infestation. So because you have all different life stages, I would go in with one miticide that will control all different life stages. Sultan is one of the options that you have, and you can follow up with ovation to clean up the eggs, and then you can follow up with a sand mite or pylon or avid just to clean up the population or the nymphs. Next, please. What about if you have a very heavy infestation and you got all different life stages? Well, in that case, same thing, because if mixed life stages, you want to start with a miticide that could control all life stages. Sultan is a good option, and then you can use avid to clean up the nymphs and the adult, flora mite and sand mite to clean up the nymphs and all the other life stages as well. So there are also a lot of other choices that you can use to control them. So all these examples are really something that I thought would work, but you can look at your own arsenal, you can look at your own storage and see what you have and kind of mix and match, thinking about what life stages they might be controlling and what is the best way to combine them. Next, please. What if you are doing biological control as well? So if you have predatory mite to control spider mite, my recommendation is if you are going to use biological control, minimize the use of any insecticides and miticide. If you don't have to use it, don't use it. If you have to use it, use the one that are the most compatible. There are quite a few compatible biological control agents listed on my handout, but over here I have Sultan, Floramite, Tetrasan, and Judo. Those are some of the good choices. Next, please. For control of broad mite and cyclone mite, well, there are not that many options out there. Pylon, Judo, Avid, Akari, Sand mite, Sirocco. Those are some of the options that we have available. Next, please. Eriophyid mites. The chemical would be Judo or Forbid if you're outdoor production. Avid, Akari, Pylon if you're in the greenhouse. Sirocco, Kelthane, Contos, wood oil, and insecticidal soap. Eriophyid mites is a pretty difficult thing to deal with, so it's always a good idea to mix in a spreader stickers with your miticide to control them. Next, please. What if you have a spider mite population mixed with thrips population? Well, then you can use a chemical that deal with both group, kill the spider mite and kill the thrips at the same time. You have Pylon, Avid, Conserve, and Sirocco. Those are some of the options. Or you can do a tank mix of a miticide and a thrips material. But when you do tank mix, make sure that they are from 2 different mode of action so that you can reduce the chance of developing insecticide resistant. Next, please. So if you have a spider mite population mixed with whiteflies, the option would be Judo, Avid, Sand mite, and Sirocco. This chemical deal with both group of pests. And other options, of course, is to tank mix a miticide with a whitefly material. Next, please. So, and that would be the end of what I got.
Wonderful, JC. Great information. We've got more questions coming in. We're going to tackle a couple of them right here. You mentioned spreader stickers. I don't know what Captiva is, but Stephen wants to know, how effective is Captiva added to a miticide?
Captiva. Stephen, do you mean capsule or was it Captiva?
Yeah, I don't know Captiva. We'll see if he weighs in on that one. A couple of people want to know about avid and resistance. They've observed a little bit in their own facilities. Have you seen that? And then Russell wants to know, are there any other translaminar miticides with similar activities and results as avid?
All right, so let me tackle the translaminar activity, that question first. There are quite a few translaminar miticide out there. One of them, of course, is Avid. The other one is Pylon. Pylon, you could use that in greenhouse only at the moment. And another one is Tetrasan. Tetrasan also have translaminar activity. Altiflora, that's by Gowan. That one is registered for nursery use production, and that one has a translaminar. Judo and Forbid as well, they have translaminar activity.
And we know what Captiva is now. It's a pepper and garlic oil.
Pepper and garlic oil. Well, I have not tried them, But
great for lunch, but
great for lunch. And years ago, when I was working with mealybugs, and we used pepper oil and one of the very similar product, pepper oil, essential oil, and it burns my eye to no end. So if you're going to use any kind of essential oil, my suggestion is to be very careful with phytotoxicity and also the fume that might be coming out from it. So I cannot say for sure what Captiva could do for spider mite population, since I have never tried that before. Sorry.
There you go, a research project. Jesse wants to know about insecticidal soap. What are your thoughts on that, and which one would you suggest?
Well, there are quite a few insecticidal soap out there. The insecticidal soap typically is pretty safe on most of the plant species. I do not typically recommend them for controlling spider mites or any kind of mite species, simply because we have so many other miticide out there. But in organic production, it's definitely one of the options that you could use. The one thing about using insecticidal soap is you need to understand it has zero residual at all, meaning that as soon as you spray, as soon as the solution dries, it has no control against any other insects or mites whatsoever. So it would obligate you to spray more frequently, and also making sure that you do your best in making sure no new invasion coming in after the applications.
All right, and last question, we're a tad over time, but that's all right because this is great information. Robert wants to know, can mites go dormant in the greenhouse over the summer?
That is
like along walkways and benches, and for how long and how hot can they handle in a greenhouse?
So that is a very interesting question. I have not researched into that particular topic. Let me think about that. If it is spider mite, typically they do not survive very well. If it's a spider mite and it's warm enough, they typically do not stop their activity. So they are just going to keep going. If they cannot find any plants to feed on, they're just going to die. But if it's a cool season mite, which actually go dormant in the hot months, there is a chance that they might be able to survive in the greenhouse. But greenhouse is also a great way to clean up your population, which is if you close all the windows, all the doors, and let it cook for a long while, you actually can kill a lot of mite species or insect eggs and overwintering stages in there.
All right, great information. Now, if you've got more questions, or if you want that fabulous handout, which I know you want, here, as promised, is JC's email address, and he'll hook you up. And certainly, if you have any other kind of questions, ask him at any time. 24/7, he is right there on the computer, ready to answer your questions. His wife loves him for that.
That's right. If you call me and you hear a grunt in the background, it's not my wife. She's used to it. It's actually my dogs.
All right. And as promised, can't say when, but by tomorrow for sure, this webinar and all these great questions and answers will be archived at ballpublishing.com/webinars. And lastly, another big thanks to our sponsor, BASF, who makes these free webinars possible. So with that, for JC, for BASF, and for all my folks at Ball Publishing, this is Chris Beytes saying, so long, everybody.